Shahabuddin, Vice Chancellor, Universiti Kebangsaan Malaysia, Mr. Uwe Morawetz, International Peace Foundation Chairman, and the Honourable Deputy Vice Chancellor of UKM. The Honourable Professor Robert Horvitz, the Honourable Professor Dr. Sharifah Hapsah Said Hassan Shahabuddin, Vice Chancellor, Universiti Kebangsaan Malaysia, Mr. Uwe Morawetz, Chairman of the International Peace Foundation, senior officials of UKM, distinguished guests, ladies and gentlemen. Good afternoon. Professor Horvitz, welcome to Universiti Kebangsaan Malaysia. It is indeed our pleasure to welcome you to our beautiful campus as part of your visit under the Malaysian Bridges Program. To begin this afternoon's program, I would like to invite you and the audience to view a short video on the activities and achievements of Universiti Kebangsaan Malaysia. Ladies and gentlemen, as mentioned earlier, Professor Horvitz is visiting Malaysia as part of the Malaysian Bridges program. This program involves the participation of Nobel laureates and is organized by the International Peace Foundation in collaboration with Malaysian universities. UKM would like to thank the International Peace Foundation for the invitation to co-host the event for the second time. I would like now to invite the chairman, Mr. Uwe Morawetz, to say a few words.
Ladies and gentlemen, friends, welcome to the second ASEAN event series, Bridges, Dialogues Towards a Culture of Peace. Bridges is facilitated by the International Peace Foundation, a non-political and non-religious foundation under the patronage of 21 Nobel Peace Prize laureates based in Vienna. The events are hosted in cooperation with the various local partners, including the country's major universities. And I would like to thank UKM and its Vice Chancellor, Professor Dr. Sharifa Hafsa Syed Hassan Shahabuddin, for hosting our event today. Having started in November 2008, Bridges is being continuously held in Malaysia and in Thailand until April 2009, involving the participation of Nobel laureates for peace, physics, chemistry, medicine, and economics. The second ASEAN series of Bridges is an independent contribution to the Decade for a Culture of Peace and Nonviolence, initiated and promoted by the United Nations General Assembly. It follows the Bridges series, which the International Peace Foundation has already hosted in Thailand and in the Philippines since 2003. The pluralistic program of Bridges highlights the International Peace Foundation's intercultural and transdisciplinary approach towards peace. The foundation doesn't take sides, but acts as a mediator by creating an independent platform for dialogue where representatives of science, politics, economy, culture, religion, the media, and youth meet, share their viewpoints, listen to each other, and find mutual ways of understanding and cooperation. I don't know if it's also the sound is also the same. Yeah. Anyway, the foundation itself is a bridge and a facilitator between different language groups in our divided societies, where politicians speak another language than artists, and business and religious leaders another one than scientists. In a highly interdependent world, problems cannot be solved either by one of these language groups only, but by working together. After the success of its Bridges program in Thailand and the Philippines, the International Peace Foundation has been invited by all ASEAN countries, with the exception of Myanmar as for now, to build further bridges towards peace and international understanding by expanding its Bridges program in Southeast Asia to stimulate the intellectual, scientific and cultural exchange in the region. The second ASEAN Bridges series therefore continuously takes place in Malaysia and Thailand from November 2008 to April 2009, comprising events with Nobel laureates from all fields. The Nobel laureates visit the region, not all at once, but separately to conduct public lectures, seminars, workshops and dialogues hosted by local institutions during a continual period of 6 months. The aim of Bridges is to facilitate and strengthen dialogue and communication between societies in Southeast Asia with their multiple cultures and faiths, as well as with peoples in other parts of the world, to promote understanding and trust. The events aim at building bridges through Nobel laureates with local universities and other institutions in Southeast Asia to establish long-term relationships which may result in common research programs and other forms of collaboration. By enhancing science, technology and education as a basis for peace and development, the events may lead to a better cooperation for the advancement of peace, freedom and security in the region with the active involvement of the young generation, ASEAN's key to the future. This is why Bridges is not designed as a one-time event, but as a continuing process of synergies to make the series of events a sustainable success for Malaysia and for Southeast Asia as a whole. I'm grateful to the Malaysian Honorary Chairman of Bridges, His Royal Highness Raja Nasrin Shah, and to our Chairman, Tumuza Hitam. Their powerful guidance paved the way for Bridges to bear fruit. I'm also grateful to the representatives of our local partner institutions who have developed the detailed schedule of events and continuous meetings, as well as to our sponsors who have enabled to make the idea of Bridges a reality. I would like to say thank you to everyone present today for taking part in this program. May it help us to facilitate a new culture of peace through dialogue, transcending its definition as merely the absence of war or armed conflict, into a new understanding what the basis for peace is: education. In this spirit, we welcome today the 2002 Nobel Laureate for Medicine, Professor H. Robert Horvitz, who has agreed to come to Malaysia to support the events. We all look forward to his keynote speech and to his important contribution to build bridges.
Thank you, Mr. Morawetz. It now gives me great pleasure to call upon Yang Berbahagia, Professor Dr. Sharifah Hapsah, Vice Chancellor of Universiti Kebangsaan Malaysia, to welcome and introduce Professor Horvitz to the audience.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera. Professor Robert Horvitz, Nobel Prize winner in medicine, year 2002. Welcome to UKM once again. And Mr. Uwe Morawetz, Chairman of the International Peace Foundation. Once again, you're coming the second time, I think, to UKM. Dev kehormat, timbalan-timbalan Chancellor, Urusan Utama Universiti, para profesor, tetamu yang hadir. Saya ucapkan bagi pihak Universiti Kebangsaan, selamat datang ke siri syarahan Nobel Laureate, Bridges Dialogue Towards a Culture of Peace, anjuran bersama International Peace Foundation, dan sekali ini akan disampaikan oleh Profesor Robert Horvitz, pemenang hadiah Nobel dalam bidang perubatan pada tahun 2002. Seterusnya, izinkan saya meneruskan ucapan dalam bahasa Inggeris. I hope you understood that. Ladies and gentlemen, Professor Horvitz is a very renowned microbiologist who shared the 2002 Nobel Prize for Medicine for discoveries in the genetic regulation of organ development and programmed cell death, or apoptosis. Please read the short biograph that you have in front of you to get a glimpse of his distinguished career and the numerous awards that illuminate his achievements. From a person who has been engrossed in basic research at the molecular level, we indeed are very privileged this afternoon to hear his views on biomedical sciences, world health and world peace. Good health, as we know, is the essential foundation for individual learning. And with education, we hope for a better quality of life, and essential ingredients for equality and world peace. From him, let us hear the connection between molecular events, health, and peace. Professor Horvitz, no doubt, will demonstrate how basic research in biology can lead to discoveries that promise new approaches to the prevention and treatment of diseases. A most striking example of the contribution of basic research is the discovery that genes, I think you know what genes are, they are so remarkably similar among different organisms that we can infer how they work in humans by studying what they do, for example, in yeast that we use to brew beer, or in a worm, or in a fruit fly. By understanding the mechanism, we can intervene to cure or prevent human diseases. Professor Horvitz worked with cells of the roundworm that lives in the soil. They're not earthworms, right? They're smaller. That work is dramatic in the discovery that fundamental biological processes are shared by animals as diverse as the roundworm and human beings. It led to the award of the Nobel Prize in Physiology or Medicine in 2002. I think I know what the roundworms are. Ascaris rhamnoides. Professor Horvitz discovered the genes that control the process of how certain cells in the roundworm commit suicide as the worm develops. However, they don't commit suicide in our elementary tribe. The worm, I mean, not the cells. Corresponding genes have subsequently been proven to control the process of cellular suicide in all animals, including humans, in a process called programmed cell death. And the misregulation of this programmed cell death has been implicated in a broad variety of human diseases, such as cancer, liver disease, heart attacks, strokes, and the neurodegenerative diseases. This understanding has provided new approaches to the development of pharmaceutical agents for the treatment of such disorders. Thank you, Professor Horvitz, for showing us the importance of basic research. At a time when we are all pressured by the utilitarian aspects of research, being hounded to produce results for application and commercialization, we thank you for underscoring the principle that basic research is the driver of new knowledge and innovation. As a national university, we believe that research should not only create wealth, new knowledge and new wealth, but also promote socio-economic well-being. I hope my faculty members and researchers, in particular our researchers at the Institute of Medical Molecular Biology, faculties of medicine, pharmacy and allied health, as well as the School of Life Sciences, are inspired by you. They would be particularly interested in your use of genetics, molecular biology, biochemistry, electrophysiology, laser microsurgery, and pharmacology to study how genes control the development of the nervous system and how the nervous system controls behavior. All these disciplines are within the reach and scope of our researchers and academics, as we've tried to show you in the video that we promote very much multidisciplinary research and collaboration. We too dream of giving birth to a Nobel laureate. Listening to you today is a step in that direction. Ladies and gentlemen, please join me in extending a very warm welcome to Professor Horvitz.
First, thank Uwe Morawetz and the International Peace Foundation for inviting me to participate in this very important program. I have learned much about this program, both before my visit and during the past day that I have been here. And I must say that it is both interesting and significant, and I'm honored to be able to be a part of it. Having looked at the previous participants in this program, and I know some of them, and at the program going forward, it is a marvelous group of highly diverse individuals, and maybe together we can all make a small step forward towards the admirable and very important goals that this program advocates. I also want to thank the National University of Malaysia, the Vice Chancellor in particular, for hosting me today, and for those kind remarks, and also for sharing the video, which I must say was eye-opening and made me ask many questions. I would like to know more details about the various topics that were raised, because there are clearly some very interesting, important areas that were being addressed in that video. I also want to say that personally, I'm very appreciative for this opportunity for both myself and my family. My wife and daughter are with me on this trip to come to visit Malaysia. None of us has ever been here before, although we feel a part of us is very much a part of this country, because we had a young woman from Malaysia live with our family for 2 years. She's from Penang and now lives in KL, and she and my daughter are off someplace this afternoon. I'm not sure where, having a good time. And we also have a young woman who's lived with us for the past 2 years, who, until she was 21, she informed me, was a dual citizen between Malaysia and Singapore, but she chose the other option at age 21 because of what she was doing, and I won't try to explain that. But I feel like I have heard over these past 4 years much about Malaysia, and the opportunity to come visit and meet many people is very special to me. Now, I've entitled my lecture today, Biomedical Science, World Health, and World Peace. And basically, I believe that these 3 topics are vitally interconnected. World peace is very much threatened by inequalities around the world, inequalities in wealth, inequalities in education, and inequalities in health. And to me, the last of these, the inequalities in health, is very much the driver and an essential component of the other 2, because without good health, neither education nor wealth in a society can be improved. And I think about this topic in the context of a conversation I had one night at dinner with Daniel Vasella, who is the CEO of the international pharmaceutical giant Novartis. And Dan was telling me that he had established a foundation, and the goal of the foundation was, in fact, to try to explore, to try to address some of the world's inequalities. And his first project was to develop, in fact, to build a school in the West African country of Mali. And Dan went to the dedication ceremony that celebrated the opening of the school, feeling very happy that he had made a big difference in the lives of a small number of young people. And what he told me was that he was very disheartened by his experience. And the reason was that it was clear the school was going to have very little impact on the lives of these children, because the children were simply too ill to take advantage of schooling. They needed help before they could become educated. And Dan said that was a profound lesson for him, and it's a lesson that I feel I want to share with you today. So what then is needed to promote world health? And I think there are 2 things that are crucially important. One is there must be, is this on?
No, you use this one.
Oh, I'm using it, that's
This
right.
is
Is this on?
yeah, this one.
And you can hear me.
Yeah.
That's good. First, there must be one or more broad international consortia that focus on global health. Such efforts must involve the active participation of governments and also of foundations, pharmaceutical companies, hospitals, and most importantly, very, very dedicated individuals, because institutions are nothing without people. So there must be these drivers to obtain and distribute appropriately financial support, and there must be the right infrastructure in each country to provide health care to all of the people in this country. In addition, there must be advances in methods for diagnosing, for treating, and for preventing disease. Many of today's diseases we simply don't know how to treat, or the treatments themselves are dangerous, or the treatments are so expensive we cannot imagine using them in any way globally. In addition, resistance to existing treatments often arises, as I think everybody here knows, for example, with malaria, which is predominantly resistant to chloroquine in this country. And that resistance requires the development and sometimes the repeated development of new treatments. So we must be prepared to make advances in these ways. And these novel advances in treatments must be driven by basic biomedical science. And in this lecture, what I really want to do is to demonstrate with one example close to my heart, namely the research of my own laboratory, with one example how basic research can lead to discoveries that promise to provide new approaches to the treatment of major human disease. I also will advocate that governments and foundations allocate a significant portion of their research portfolios to basic research, despite the fact that in many countries what is needed is specific applications of basic knowledge for particular problems. I think it is nonetheless important, vitally important, that the basic research be advocated and supported, and I will tell you why I believe that. Now, in a more general sense, I'm going to start by telling you about what I believe is one of the most striking discoveries in the history of science. And that's something we've just heard a few words about, namely genes. And genes are those bits of DNA that make us what we are. Genes are so remarkably similar amongst different organisms that we can study what they do in any of a variety of creatures, like yeast, like a roundworm, like a fruit fly, like a zebrafish, like a mouse. Any of these organisms we can study and learn something about genes that are in us, and genes that are important for human biology, and genes that, if they go wrong in human biology, can lead to human disease. And from these studies of genes in these simple organisms, learn about how we can intervene with their functions or dysfunctions, and in that way approach novel cures for human disease. Now, my own research mostly involves, as you heard, a microscopic roundworm, a worm known as C. elegans. Elegans because it is elegant. It is beautiful. Now, you may not think worms are beautiful, but beauty is, as has been said, in the eye of the beholder. And I will persuade you that this is a beautiful animal. I'll focus on the 2 lines of research using this tiny worm that resulted in my sharing the Nobel Prize in Physiology or Medicine in 2002. Now, a word about the Nobel Prize. Many people know that it comes with a gold medal, and it also comes with some money. What I didn't know was that it comes with a diploma. So this is my Nobel diploma. And if you read this diploma, assuming you can read Swedish, you'll see that at the end, the citation says that this prize was for the understanding of the genetic control of organ development and programmed cell death. So what does this really mean, and why did I receive a Nobel Prize for studying a tiny millimeter-long worm? Okay, first let me start with organ development. Okay, organ development refers to our studies of the development of the major female genitalia of the worm. And I think the Nobel Committee decided to use the word organ development so they didn't have to go into any more detail. I will not go into any more detail either at this moment. There we go. These are pictures of the worms. Now, these worms come in two sexes. The bottom animal is a male. The top animal is not. It's a hermaphrodite, and in fact, it produces both sperm and egg, and can be completely self-sufficient reproductively. It can also mate with males and produce offspring as a result of mating. These features turn out to be very advantageous for studies of genetics, and I'll turn to the area of genetics in a few minutes. Now, what we studied was the development of the sexual organ of the worm, the vulva. You can see the red line there. And this is the organ that is used for egg laying, so eggs come out of the uterus and are deposited into the environment, and also used for mating. So now, how can our studies of this organ have led to, in fact, fundamental breakthroughs in cancer biology? So let's talk about cancer. What causes human cancer? The basic cause of human cancers is variants of genes. And again, I'll say a little bit more about genes a bit later. The first human cancer gene was identified in 1981. This gene is called RAS, and RAS has proved to be a major gene in many human cancers, including lung cancer, pancreatic cancer, bladder cancer, leukemia, neuroblastoma. This is a major cancer gene. So this gene was discovered. It was clear that it was involved in human cancer, and people, scientists, desperately wanted to know, what does this gene do? And what's the pathway in which this gene acts? And let me say a little bit then about pathways and gene pathways. Pathways are key for understanding biological processes, and pathways are key, biological pathways, for developing drugs. Why is that? Well, if you have a gene and something's wrong with it and it causes disease, how would you think about trying to eradicate that disease? Well, what you might do is you might eliminate the function of the disease gene, and if you do that, you get rid of the disease. Okay, that's a very simple approach to disease. But now, what if you know that that disease gene causes disease not in a single step? Our bodies are very complicated. What happens, in fact, is that disease gene starts a process, and it has an effect, and then that next step has another effect, and so on and so forth. So there are many genes that can be between a disease gene and the ultimate disease outcome. And if you block any of the steps in this pathway, you will block the effect of the disease gene. So by understanding genetic pathways, gene pathways, one, you understand basic biology, and two is you identify many more target sites, target genes, at which you might intervene in a disease process. So one of the major problems in modern biology is to identify the pathways through which genes, both healthy genes and disease genes, function. So, having said that, what about RAS? What about the RAS pathway? The answer emerged not from studies of human RAS, even though human RAS had been identified as a cancer gene. The pathway emerged from very basic and initially apparently totally unrelated studies in the field of developmental biology. And the progress really came from two studies. One was our studies of worm vulva development, and the other was from studies of the eye of a very small fruit fly, Drosophila melanogaster. And it turned out in both of these aspects of development, a gene just like the human RAS cancer gene was involved in normal development. The genes were so similar that 80% of the gene was identical between among worm, fly, and human. And what that meant was that if one studied what these genes did in these other organisms, in these very small and very simple organisms, one could in fact figure out more generally how the gene worked, and it turned out that how it worked was exactly the same in the three organisms. So these studies, which were done by my lab at MIT, by one of my ex-graduate students' labs at the California Institute of Technology, and in terms of the fly eye at Berkeley in the United States, these studies led to the identification of an entire pathway in which RAS functions. And this pathway turns out to be a pathway of signaling between cells. One cell releases a signal that is received by a molecule called a receptor on another cell, which then signals another protein made by a gene to another one, then getting to RAS, which in turn signals 1, 2, 3, 4 more steps later, and ultimately leads to a biological output. That pathway is responsible for development of the worm vulva, it's responsible for the development of the fly eye, and it is responsible for many, many human cancers. For example, when RAS, this cancer gene, is mutated to cause cancer, what happens is there is a change in this gene that makes the RAS protein active even when there's no signal. So it's behaving as if there is a signal when there is none, and that makes it drive cells to divide when normally it wouldn't be doing so, and those extra dividing cells are what lead to the cancerous state. Now, what good is the knowledge of this pathway? Well, based on this pathway, many drugs have been identified. For example, here are 3 drugs currently being clinically used that affect the RAS pathway, and they do so by affecting the molecule that responds to the signal from another cell. And they basically prevent that signal from activating this pathway when it shouldn't. Okay? And these are drugs used in cancer biology. But if you think about it, from what I've told you, if RAS, when it's activated, starts this pathway, it's going to do so after the point at which these drugs work. So knowing about the RAS pathway has been very important for treating. It doesn't really cure, but it improves cancers that involve this pathway, but not those cancers that involve mutations in RAS itself, because those mutations will bypass the effects of those drugs. So we get 2 things out of this. One, we get the pathway with a number of targets which are effective in a variety of cancers, And two, we now understand why those very same drugs are not good for all of the cancers that involve these pathways. And we have new targets from this study that people are exploring now with other drugs that will act downstream instead of upstream of RAS. So it's the understanding of this pathway, which emerged from basic studies of a worm sexual organ in a fly eye, that led us to novel drugs and to current approaches to still further drugs for cancer. So that's example one. Let me turn to example two, from organ development to programmed cell death. And again, the phrase on my diploma says, the genetic regulation of programmed cell death. And so let me translate that. I know some of you are sophisticated biologists, and so what I say for the next few minutes will be very trivial, and I know some people in the audience know less about biology, so I will not apologize too much for the basic description. So let's start about genetics. What's really genetics in the cell? You will say cell death, what's a cell? And most importantly, what is programmed cell death? And why should anybody care about dying cells? First, genetics. Genetics is the study of genes. Genes are responsible for all the biological processes that occur in living organisms. And genes are the basis of heredity. We each inherit half of our genes from our mother, half of our genes from our father. Variations in I told you all of that, so we'll keep going. Variations in genes amongst individuals lead to variations in the biological processes that these genes control. So variations in traits. So that it's differences in genes that make us different from orangutans or fish or seaweed. That's all genes. Other variations in genes have more subtle effects. They can make us slightly different from each other in eye color or blood type or whatever. And still other variations in genes can cause dwarfism or color blindness or deafness. And some variations in genes cause marked diseases. Huntington's disease, for example, is caused by variations in one particular disease. Variations in genes can also predispose us to a variety of diseases: cancer, cardiovascular disorders, asthma, cystic fibrosis, premature aging, Alzheimer's disease, bone loss, many disorders we can be predisposed to, i.e. more likely for us to get these diseases because of particular gene variants that we carry, but we can't be sure that that's going to happen, and it may depend upon some combination of our other genes and also upon the environment to which we are exposed. So genes are important to us, they're crucial to our health, and then it becomes very important to study genes and understand what they do. And one approach is to study human genes. Many biologists do this. In fact, I also study human genes in addition to studying worms. But there's a difficulty in studying human genes. In many ways, the study of humans, we're complicated. And the biology is complicated. And there's some types of studies that one wants to do with genes that you can't do with humans. For example, you'd like to make combinations of genes in new ways, and to do that, you do crossings. Well, I can cross two worms easily, but if I'm looking at two human genes, I'm not quite going to be able to cross two people for a variety of reasons. That doesn't work. So we need to draw upon modern biology to study genes in other ways, and taking advantage of the conservation of genes amongst organisms is one way in which this can be done using experimental organisms of the sort that I alluded to before. And the organism that we use, again, is this roundworm, Caenorhabditis elegans, C. elegans for short. And that's been the primary organism I worked on, and I'll turn back to C. elegans in a moment. Okay, so that's a little bit about genes and genetics, and what about programmed cell death? And starting with what is a cell? Well, in short, a cell is the fundamental unit of life. Our bodies are made up of cells. We have skin cells and nerve cells and muscle cells and blood cells. So cells are the unit of life. What do we mean by programmed cell death? Well, cell death is easy. Cells are alive, and if they're not, cells are dead. Programmed cell death. By programmed cell death, what I mean is naturally occurring cell death. Cell death that occurs in organisms as a normal part of their biology. And that's very counterintuitive, because we think about organisms as wanting to keep cells alive. But it turns out that in many cases, normal biology makes cells die. And let me explain how that comes about. Think about development. Okay, we start with a single cell, a fertilized egg, and that cell divides and divides and divides, making more cells. And for example, in us, it can make as many as 10 to the 13 cells, 10 million cells. And each of these cells must do something special, must take on special characteristics, become skin or blood or nerve or muscle or whatever. And these cells must sometimes move around from one place to another, and then they must aggregate in the right ways to make structures like fingers and toes and so on. Or even more complicated structures like our brains with all the right interconnections of the nerve cells so that the brains are capable of doing what they do. These processes, cell division, cell differentiation, cell migration, morphogenesis, these are the basic properties and the basic problems of developmental biology. It turns out there's another basic process that appears to be universal among developing animals, and that's the process of cell death. Quite remarkably, many of the cells that are generated as animals develop, and that includes us, do not survive, but instead they die. It's this naturally occurring cell death that is referred to as programmed cell death. It had been known for some years that cell death occurs during the normal course of animal development, and you're all familiar with some examples. Think about a tadpole developing into a frog. Okay? The tadpole loses its tail, and the cells in its tail die. That is programmed cell death. If you have studied medicine or developmental biology, you know that in utero, a human baby has webbing between its digits, its fingers and its toes. And that webbing is removed in utero before birth. That webbing consists of cells. Those cells undergo programmed cell death. Think about birds. Think about bird feet. Bird feet come in two varieties: webbed, good for swimming, and not webbed, good for walking. The difference between these two kinds of birds has to do with whether or not programmed cell death occurs to get rid of the webbing. Programmed cell death can be a major event. In areas of the developing mammalian brain, as many as 85% of the nerve cells die. In our immune systems, as we sit here today, in our blood, the thymocytes, a particular kind of blood cell, as many as 95% of those thymocytes we are generating today will die by programmed cell death. So this is a very widespread phenomenon. Nonetheless, this phenomenon was ignored by biologists for many years. And I think one reason was simply thinking about it. Biologists, they couldn't think of cell death as being something that organisms wanted to do. If a cell dies, that's because the cell has been mistreated. And that was about as far as the thinking really went. And it was our discovery that this view is wrong, I think, that really led to the Nobel Prize. Because what we discovered was that there is a biology of cell death, and that cell death is an active process, meaning that there are particular genes that act to make cells die, and furthermore, those genes act within the dying cells, so that the process of programmed cell death is actually a process of cellular suicide. And I should say I got into a very complicated discussion one time with a member of the church in Switzerland, and talking about the philosophical implications of the fact that normal biology has a suicide process as a part of the establishment of a living human being. Okay? I won't go into all of that today. So there's a biology of cell death, and it's every bit of as much of a biology as there is a biology of other cellular processes like cell division, cell migration, and cell differentiation. Now, any biological process in us, if it goes wrong, can lead to disease. And thinking now about cell death as a biological process, it became clear that programmed cell death is no exception to that. And there are many diseases that are today recognized to be diseases of abnormalities in programmed cell death. First and easiest to see are disorders in which cells that should live instead die. And you can imagine then that it's simple that that would lead to disease. Neurodegenerative diseases, Alzheimer's, Huntington's, Parkinson's, amyotrophic lateral sclerosis, and about 24 others. Nerve cells that should live die, and our nervous systems fail to work the way they should. But in addition, stroke, traumatic brain injury, which in many countries means falling off a motorcycle onto your head, AIDS, heart attacks, congestive heart failure, liver injury, many liver diseases, including those caused by the hepatitis C virus, and many other disorders. Cells die, and it turns out that the way in which those deaths occur use the same mechanisms that are used for programmed cell death. This normally dormant process of cellular suicide becomes unleashed, and the wrong cells are at the wrong time, and cells that should live commit suicide and die. There are also disorders of the opposite sort, disorders in which cells that should die instead live. And that can lead to disease as well. And it turns out cancer is one such disease. We think about cancer as being too much cell division, and that's accurate, but it's only half the story. If you think about a tissue in the human body, the number of cells in that tissue, if that tissue is a dynamic tissue, the number of cells is going to be defined by two opposing processes. The rate of cell division, which is always adding new cells, and the rate of cell death, which is subtracting cells. If you have too much cell division, you'll end up with a cell proliferation. If you have too little cell death, you also will increase cell number. And it turns out that many, and probably all cancers, involve both of these phenomena, too much cell division and too little cell death. And certain cancers, such as follicular lymphoma, which is pictured in the slide, certain cancers are fundamentally driven by too little programmed cell death. So this means that an understanding of the process of programmed cell death should lead to an identification, both an understanding of disease and an identification of genes and gene products, proteins, that might be affected therapeutically to make improvements and to develop treatments for a broad variety of human diseases. And it was really our contributions to this understanding that were recognized by the Nobel Prize. Now, as I said before, we analyzed the process of programmed cell death not by studying it in humans or even in another mammal like a mouse, but rather by studying this tiny little, very beautiful worm. C. elegans played the major central role in the Nobel Prize, and I had two co-recipients, both of whom were my colleagues for some years, Sidney Brenner and John Sulston, who were honored because of their contributions to this work. So what did they do? This is Sidney, and he introduced C. elegans to modern biology. He worked out the basic methods for its study, and he was drawn to this animal in large part because of its simplicity. You'll recall that I said that our bodies have something like 100 million cells, and if you take an insect, you probably have, depending upon the insect, between 100,000 and 1 million cells. That's a lot simpler. C. elegans has 959. Okay. That's it. 959 somatic main body cells. Much simpler. In addition, Sidney Brenner was drawn to C. elegans because it's highly suitable for studies of genetics. I won't go into the details about why that's the case, but it's a perfect organism for genetic studies. and he was able to ascertain the complete pattern of cell divisions that generate this animal. So again, we start with a single cell day, and it divides to make 2 cells. Well, this shows you that it doesn't just go 1, 2, 4, 8, 16, 32. But in fact, the pattern of divisions is more complicated and much more interesting. And the pattern of cell fates that emerges allows you to begin to think about what processes might be going on as an animal develops. John worked out the complete cell lineage of this animal and determined how, starting with a single cell, the 959 cells in the body are generated. In the course of this work, he discovered that the animal generated more than 959 cells. There were 131 additional cells that were made that are not found in the adult animal. And the reason, as you can probably guess by now, is that these 131 cells undergo programmed cell death. So Sidney worked out the basics for the worm. John discovered 131 cells undergo programmed cell death during C. elegans development. And then I and my laboratory worked out the identification of the genes and processes that were involved in these cell deaths. And the way in which we did this is we looked for mutant worms, genetically abnormal worms, in which the process of cell death was altered. For example, if we look at the top image in this slide, we can see animals. These are the heads of animals, and you can see these flat disc-like appearances. These are programmed cell deaths. Look at the bottom animal, and you don't see any of those. This is a mutant in a gene called SED3, CED for cell death abnormal. And in a SED3 mutant, it turns out none of the cells that should undergo programmed cell death die. The SED3 mutant has this SED3 gene inactivated. So when this gene is turned off, cell death doesn't occur. What does SED3 do? Well, it's very simple. SED3 is a killer. SED3 drives the process of programmed cell death. So this discovery said that there is a gene in the worm necessary for the process of programmed cell death. Hence, programmed cell death is an active biological process. It requires a gene that is encoded in the DNA of this organism. We then found a second killer gene, called it SED4, and we showed that these 2 genes work within the cells that are going to die to cause them to die. And these were the observations that led us to conclude that programmed cell death, at least to this extent, is a process of cellular suicide. These cells are killing themselves. We then found the third gene, SED9. I won't bother telling you about 1, 2, and then 5, 6, 7, 8. They exist and they're related, but I'm not going to go into them today. SED9. SED9 is an anti-death gene. SED9 protects cells from undergoing programmed cell death. And then we found another killer gene, AO1, which just like SED3 and SED4 causes cells to die. Turn it off, everybody survives. So we had 3 killers and a protector, and I won't say what that numerology might imply about life on Earth, but that's what we had. And we put them into a pathway. Using standard genetic and molecular genetic methods, we could see that SED3 kills. If SED3 is active, a cell is going to die. SED3 is activated by SED4. So SED4 kills by activating SED3. SED9 protects by preventing SED4 from activating SED3. And EGL1 kills by preventing SED9 from preventing SED4 from activating SED3. And that is the core pathway for programmed cell death in CLAs. Now, we analyzed these genes and discovered that SED9, for example, looks like a human cancer gene. It's a gene called BCL2. BCL2 is named for B-cell lymphoma. This is a gene I alluded to before because this gene, when mutated in the appropriate or inappropriate way, is responsible for follicular lymphoma. So SED9 looks like a human cancer gene. And we found that these genes are so similar that if we took the human gene and put it into a worm, it could protect against cell death, and furthermore, it could substitute for the worm gene. So the fact that the human gene could work and substitute in the worm said that not only these two genes must be parallel, but there must be similar pathways in these two organisms, worms and humans. And pathways, again, are important. We studied the molecular nature of the other genes. SED3 turned out to look like a human protein called ICE, interleukin-1 beta converting enzyme. Now actually, when we discovered SED3, it looked like nothing, but we thought that was too boring to publish. So we waited until a gene emerged in the database that looked like SED3, and it emerged from studies in a pharmaceutical company interested in human inflammatory disease. And when we saw the similarity of SED3 to this human gene, something was known about how the human gene worked, and that gave us a very strong clue about how the worm gene worked. But the day we published this paper, I was contacted by 5 pharmaceutical companies asking me about how this human gene might relate to cell death, because that's not what they thought they were studying. And it turns out, well, it does. In fact, there's a family of about a dozen of these genes today known as caspases that are present in humans, and most of them have been shown to work in us to drive programmed cell death. And SED3 and this gene ICE are the founding caspases. SED4 also has a human counterpart. It was discovered some years later in studies of programmed cell death in human extracts. And EGL1 turns out to be similar to a series of human genes that are part of the BCL2 superfamily, again, this oncogene I referred to before. So all in all, the pathway of genes that we identified in the worm, all of these genes have human counterparts, and the studies that have been done in humans have indicated that the core pathway for programmed cell death, the pathway that is so fundamental in development and is also so important in a broad variety of diseases, is essentially the same between worm and human. Now, every cell in the animal, that means a worm, that means us, has to decide whether it should live or die by programmed cell death. In addition, so we have this first step of decision. Every cell must decide whether or not to live. Once a cell decides that it's going to die, we have literally the execution step, and that's where these genes EGL1, SED9, SED4, SED3 work. And then there's another step in programmed cell death. We don't leave the cell corpses in the body of the animal. They get swallowed by other cells and degraded. So there are 4 steps in this process: a decision step, a killing step, an engulfment step, and a degradation step. And if that's a little bit too complicated, we can think about it using some other language about what's going on during this process. This is the essence of programmed cell death. Identify the victim, kill, get rid of the corpse, and destroy the evidence, the debris in the dying cell. Okay, so again, here's the overall pathway. I haven't talked, you don't want me to talk, about all of the genes, and this isn't even all of the genes that we've identified. But the bottom line take home is, starting with these simple studies, we've identified in detail the process of programmed cell death. Now, how do we think about that in the context of therapeutics? Well, since these genes have human counterparts, intervening with the human counterparts offers the prospect of intervening with the diseases that are mediated by those counterparts. So that, for example, caspases, SED3. Caspases kill. If we could block the activity of a caspase, okay, then we could prevent illnesses. We could treat illnesses in which there is too much cell death, like neurodegenerative disorders, heart attacks, heart failure, liver diseases, and so on. On the other hand, if we can block the protective genes, like SED9 and BCL2, this cancer gene causes cancer because it protects cells that shouldn't be protected. If we could prevent the protective function, we could reactivate the cell death and cause cells to die that were supposed to die, but because of the disease process instead survive. If we could treat disorders like cancer and autoimmune disease. So based upon this thinking, many companies, big pharmaceutical companies, smaller biotechnology companies, are developing drugs against these targets. And personally, I can say I've been involved in both the pharmaceutical and biotechnology industries. I founded a company based upon the biology of programmed cell death. My company developed an antagonist to the BCL2 family of protectors, which is now in clinical trials in the hands of a major pharmaceutical company in the US. And we also developed for cancer, so we have a drug that's being now tested in people for cancer, and I would say it looks promising, and I have no financial interest in it anymore. And we also have a drug that we developed to inhibit caspases that is in the clinic, in fact, in late phase 3 trials in the clinic, that's the end of clinical trials, for liver diseases, including hepatitis C virus. And that's, again, in the hands of a major pharmaceutical company. And I'm hopeful that these two areas and these two drugs, and also drugs being developed by a variety of other companies, will intervene in a wide variety of human diseases. Okay, so having said that, what I'd like to do now is to put the discoveries I've discussed into a broader context. The work I've described involves absolutely basic research. When I began, neither the generality nor the application of our efforts was at all clear. C. elegans, in fact, at that point was an obscure organism even to biologists. I was advised not to work on it because it would kill my career. Genetic studies are often highly abstract. I didn't target any disease. I didn't start out to cure cancer. I started out to explore the unknown. I didn't know if what we found would be relevant to any organism other than C. elegans. And what happened instead was that our studies established mechanisms that appear to be universal among animals, and our findings may well help provide the basis for new treatments for a broad variety of diseases. So I think there's a very important message here. Basic research, research that is discovery-based, will lead not only to intellectually stimulating findings, and that's important, but also to insights of major practical knowledge. Basic research is the driver of scientific, and in this context, biomedical knowledge. So now, how can basic research be supported? True basic research cannot be supported by the private sector. A company can't know ahead of time where discovery-based research is going to go. I didn't know we were going to learn something about liver disease. And no company can have a business plan that is focused with no timeline and no direction. So it has to be governments and foundations that support basic research, because the basic research is key. Unexpected discoveries are what's going to drive our future. But it has to come from a governmental or foundation base where the interests are sufficiently broad that any direction of importance will be of relevance. And I would argue that even countries with relatively small research budgets should support pure basic research, because such research is crucial in ensuring that all of the research that goes on, including the applied science that's directed towards pressing problems of immediate need, it's important to have the basic science foundation for critical, creative, high-quality, rigorous science in a society. And only with such basic research can biomedical scientists make the discoveries that are going to lead to the novel pharmaceuticals that will improve world health and help us, again, going back to the first topic, on the road to world peace. There's another aspect of the relationship between science and government that is crucial, I think, in a broad sense, in informing government policy. Government policies must be informed by good science. All too often, scientific data are ignored, distorted, or in the worst cases, fabricated to serve a political agenda. Public policies instead, I think, should be founded on the soundest science available, because it's only in this way that good decisions can be made for a country and for the world. And unfortunately, in my view, many countries have at times ignored this principle. And I will say even the US has ignored this principle. Last week, literally 1 week ago today, I had the enormous privilege of being at the White House with President Obama when President Obama signed an executive order that did two things. First of all, he rescinded a political agenda-based rule that prohibited aspects of stem cell research in the US. More fundamentally, what he did last week was to direct White House staff to develop a strategy to restore scientific integrity to government decision-making. That order is enormously important. It's an important step for the US. It's an important step, I believe, for the world, and I hope that other world governments will take note. And like the US is doing today, use scientific knowledge in making the best decisions for policies concerning energy, security, and health. And in this way, make decisions that should lead us into a world with the best prospects for world prosperity, world health, and world peace. So with that, I'm going to end with a thank you to the members of my laboratory over the past 31 years who have been involved in the science I described and science I didn't describe, and also a thank you to you for being here to share this day with me. Thank you very much.
Thank you, Professor Horvitz, for the challenging discourse on basic research as the driver of scientific knowledge leading to discoveries that will result in improved world health and subsequently world peace. We will now have a short question and answer session, and I cordially invite Dr. Sharifah to chair the session.
Thank you very much, Mr. Horvitz. Would you prefer to stand there?
Either way, I can come sit down. Fine.
I'll open the floor now to discussion. We'll take maybe 3 questions and then you can respond to that. And we'll go on and report it. You can start anyway. You can start with the big agenda about funding, about how policies should be informed by science, or you can go molecular, anyway, right?
I can't promise answers, but I'm happy to respond.
If I may start, Dr. Sharifah.
My name is Najib.
Your name?
Najib.
So, Najib, 1 minute. I think I have one question in front, and we'll go to you.
Where is the voice?
What I want to ask you is, are your cells, Professor, the same as mine, or are they different? When it comes to the micro level, are your cells and my cells different? Are they same?
So,
what is the actual number of cells in our body at this time, on average? I know it's some huge figure, but do we know exactly how many cells there are in the human body? This term cellular suicide seems rather odd, I mean, cells die, natural deaths, I think. They don't, I don't know what this term suicide really means. I think you tried to explain it, but it's a little bit beyond me.
Let me take the 3 questions before we go on, because I won't remember the first one.
Thank you.
Yeah, Najib.
Okay, so...
Just one more, because I think you have only 2 questions.
Well, let me answer those questions, if I can do that, and then we'll continue from there. So, are your cells like my cells? And the answer is yes and no. So, first order, they are remarkably similar. All of the major components are going to be much the same. However, there will be variations, and these are predominantly variations that will be defined by very slight differences in our genes. Those variations cause the cells primarily to look the same, but they can make the cells sufficiently different that there can be aspects of us as organisms and aspects of ourselves as individuals that are different. So that, for example, if we were, I'm thinking about last week in stem cells, if we're thinking about using cells in transplantation, if we used your cells to treat you for a disease, your body would recognize those cells as your own and would keep them and would maintain them. But if we used my cells, the chances are very high that your body would say those cells are foreign and would reject them. So when transplantations are done, our immune systems are capable of distinguishing self from non-self. And our cells are sufficiently different that so for the vast majority of us, somebody else's cells would be recognized biologically as different. And in fact, there are more differences than that. So that's question 1. Question 2, how many cells are in us? And the answer is, whereas with C. elegans, I can say there are 959, we cannot give a precise number for people for 2 reasons. One is, no one would be able to count it today, given current technologies. And 2 is, we are much more variable, individual to individual, and even over time, than is this microscopic worm. The estimate of the number of cells, for example, in our brains is on the order of 10 to the 12th, plus or minus. And in our bodies, probably 10 to the 13th. But even those are very crude numbers. Question 3, what is meant by cellular suicide? And what's meant by that is something very specific. And that is different from cells dying sort of nondescriptly, either because they've been damaged or because something happens over time that makes them incapable of surviving, although even this latter situation is more complicated and we might come back to it. What I mean is you take a cell that is healthy and that is capable of surviving, and you activate a program within it in which certain genes become active, and those genes make the cell die. If you could prevent those genes from being active, that cell wouldn't die. So there is an active process within the cell that causes the cell to die. And that's what I'm alluding to when I describe it as a cellular suicide. The cell is quite literally killing itself using its genes to make itself die.
What is it that distinguishes between good and bad science?
What is it that is
distinguishes
good and bad science? There's probably not a dichotomy, but rather a continuum, and it may not even be unidimensional. But good science is science that is rigorous. So there is a design, an experimental design that allows you to basically interpret your results. There are control experiments that will allow you to rule out alternative interpretations that are consistent with your results. And good science is used in a way that does not have bias. Now, many scientists may have biases about what they expect in an experiment, but those biases should not override the data that they observe. So good science is data-driven, not hypothesis per se. Let the data tell you what the answer is.
That picture was on television, but we didn't recognize you until today.
No. That was quite an event, I would say.
Why were you chosen, if I may ask?
Okay, so why was I on stage and at that point the closest person to the president? And I'm not sure I know the answer to that, but I'll tell you the best reasons I can come to. One is that I've been involved in stem cell policy in the United States. So there was a committee set up by the National Academy of Sciences to formulate guidelines for the use of stem cells. Because the U.S. federal government was prohibited from many aspects of stem cell science, most of the stem cell research, with human embryonic stem cells to be precise, most of the stem cell research that was going on in the United States was going on with no federal support. In fact, worse than that, you couldn't even use a room that was air-conditioned using overhead money from the federal government for an experiment involving stem cells. So it was going on outside the federal government. It was going on